Good afternoon. Thank you so much for joining us on this virtual Maternal and Child Health Research Institute monthly seminar series. Very exciting presentations of up-to-date latest research findings for maternal and child health. And today's speaker is Desiree LaBeaud, which I'll get to in a second. But first, a few updates and announcements that I think will be very useful. So we're very excited about December's seminar series. I wanted to advertise to you is Melissa Bondi, who's the chair of the Department of Epidemiology and Population Health, recently came to Stanford. Really exciting talk on pediatric, adolescent, and young brain cancer, Monday, December 7th. Please don't miss this. She is an amazing communicator and really has some really wonderful things to share with us. That's in December. In November, we have, next slide, the virtual Stanford Maternal and Child Health Research Institute Annual Symposium. And as this year, it will be virtual, but really features really exciting, cutting-edge topics in maternal and child health. That includes our guest speaker and keynote speaker, David Williams, the Senior Vice President and Chief Scientific Officer of Boston Children's, and a segment on a new precision health and cell and gene therapy. So please don't miss that. But also three other exciting maternal and child health sessions, including scientific advances in diversity, health equity, and social justice. Really, basically the methods for how to study this important and emerging area, as well as a whole session on COVID-19 and pandemic innovations in school-age learning, including talks from Bonnie Maldonado and Lee Sanders, Jason Wang, really I think cutting edge innovators in school-age learning, very relevant topic in today's world. And then also groundbreaking research and rising scientists abstracts, which I think really highlight very recent findings in this field. So please register for that on November 6th. As well as the week-long viewing of the posters. Please register for that now. Next slide. This is our education committee who have selected the seminar series as well as have put together the exciting symposium. We thank them for their service, and also if you have any ideas or if you'd like to be a part of the committee, please contact Roxana Van Norman. But today, and also after today's talk, we're going to send you an online survey form. We appreciate any comments, suggestions, speaker suggestions, or format suggestions which you have. So please fill it out and send it back to us. Today, we have Desiree LaBeaud, who's a professor of pediatrics, infectious disease, and also by courtesy, the Woods Hole Institute for Environment, as well as Health Policy and Research. Desiree is really a leader in climate change and epidemiology. Her lab is here at Stanford, but also has labs in the field in Kenya, Grenada, and Brazil. And really we'll talk today about the work that she's doing on vector-borne diseases and the effect of climate change. This really is a relevant topic given how climate change has affected us in the Bay Area, but are now around the world, and now how basically diseases and vector-borne diseases that we're dealing with in the pandemic. This is really a two-for-one that Desiree will present to you today. So Desiree, thanks for joining us. I just wanted to mention that there's a Q&A session there, afterwards, which I'll host. And please post your questions in the Q&A part of the Zoom. So Desiree, thanks for joining us.
Thank you so much. Can you hear me okay?
Can.
Okay, wonderful. All right. Good afternoon, everyone. It's my pleasure to be here today to present this MCHRI seminar. I just wanted to start by thanking MCHRI for all the support that they've given me over my last 6 years at Stanford, and also many of my mentees. MCHRI funding has allowed me to continue some of my field sites, the field work that we're doing in Kenya and also in Grenada, Today I'm going to present some of this data and this research, and I will definitely make sure to highlight MCHRI when I present that work. So I'm here today to really talk to you about the impact that climate change has on vector-borne diseases. And I'm going to try to highlight some of the complex environmental factors that really allow for vector-borne disease emergence in specific locations and specific times on our planet. I was asked when giving this seminar, how is this important for maternal child health? Well, I think climate change impacts all of us. And so I'm hoping that you're interested in this topic and that we can learn some together today. So my objectives for this talk are first to share with you the mechanisms by which climate change can promote the emergence of vector-borne diseases, to illustrate the association of temperature change on specific risks for specific diseases, including malaria and dengue, that we sort of use as case studies for vector-borne disease using our clinic-based surveillance that we're doing in Kenya right now. And then in the end, I'm going to talk to you a little bit about forecasting risk. So forecasting disease incidence based on these climate inputs. So, as many of you know, mosquitoes are the most deadly animal in the world. And why is this? How is it that mosquitoes kill about 1 million people in our world each year? It's because, of course, mosquitoes carry pathogens. And these vector-borne disease pathogens cause a lot of problems, they cause a lot of deaths and a lot of disability in the world. Now, I'm actually an arbovirologist. I'm a pediatric infectious disease physician, but I study mosquito-borne viruses mainly. And mosquito-borne viruses, or arboviruses, stands for arthropod-borne virus. So this is a subset of vector-borne diseases. Malaria would be a parasitic vector-borne disease. I'm talking about viral vector-borne diseases. And so these diseases require a blood-sucking arthropod to complete their life cycle. Many of them are zoonotic, so they cause problems in animal populations along with human populations. And we know of at least 500 viruses that are important. They come from 8 viral families and are quite diverse. And I've just put up here on this slide, I don't know if you can see my pointer, but this is the dengue life cycle, this is the West Nile virus life cycle, and this is the Rift Valley fever virus life cycle. And you can see that the cycles are different. They use different mosquitoes, they're different vertebrate hosts, different amplifying hosts and such. And so these viruses are very interesting and very important to study and quite diverse. Now, I'm going to talk about 2 viruses in particular today, so I just wanted to give a little brief background of dengue and chikungunya before I go much further. So dengue virus is actually the most common mosquito-borne virus in the world. Dengue impacts and causes infections in about 400 million people worldwide each year. And these infections are caused by 4 distinct virus serotypes, dengue 1, 2, 3, and 4. The way people get dengue virus is by getting bitten by an infected Aedes mosquito, often Aedes aegypti. And usually what happens a few days after you get this infected bite is that really quickly you have an abrupt onset of very high fever. Sometimes you have a rash and you can have headaches, especially behind the eye headaches, general body aches. Dengue is called breakbone fever because you get these feelings like these intense body aches that your bones are breaking inside. And then in some, unfortunately, progression of dengue virus occurs. And once the resolution of fever happens, you actually end up with a lot of capillary leak, and you can have shock and hemorrhage and bleeding as depicted here in this picture. And that actually leads to most of the dengue deaths in the world. Unfortunately, we don't have a widely effective vaccine available currently. And we don't have any drugs to treat dengue. And I just want to bring up, I put up this map here just to show you, these are the 10-degree isotherms in the world. And so between the 10-degree isotherms is where the tropics are in the world. And over half of the world's population actually lives in this area between these 2 isotherms and are at risk for dengue and chikungunya and the infections that I'm talking about today. So what about chikungunya? We can see here, this is just a recent map just showing all of the areas where chikungunya has been transmitted. And chikungunya looks a lot like dengue, right? It's another vector-borne disease. You get it after the same mosquito bite, often Aedes aegypti bite. You get high fevers that come on abruptly. And with chikungunya, one of the more pathognomonic, I guess, findings, although there is a lot of overlap between these infections, is that you can have really severe arthritis or arthralgia of many joints. And unfortunately, these joint symptoms can last weeks, if not years after your infection. And so chikungunya is actually causing a lot of long-term morbidity in regions where there's been a lot of transmission. Okay, so getting to our question, how will climate change affect vector-borne disease transmission? Well, it's a very nuanced question, and it's very complex and complicated. And so I'm hoping that by the end of today's talk, you guys will have a better understanding of how this works. So I just want to start off by saying that clearly climate change is going to have a big impact on vector-borne diseases because the vectors themselves, the mosquitoes in this case, are intrinsically temperature dependent, right? All disease vectors are ectotherms, right? Mosquitoes are ectotherms, which means they can't, unlike us, they can't create their own body heat and keep themselves warm, right? They depend on the ambient temperature outside to keep them warm and allow their biological processes to happen. So clearly, if the temperature is changing, this is going to mean big changes for the vectors and for everything that's going on inside the vectors. And when we talk about climate change, there's the overarching climate change, but then there's also global warming, right? And I'm trying to see, what we're looking at here is this very cool graphic from the Washington Post. And what it's showing us is 5-year increments of the temperature in the world, starting in the 1800s and then ending in 2018. And as you see the globe turn red, and here's 2018, where you see a lot of the globe is dark red. What the dark red shows is that there's been a 2-degree increase, 2 Celsius increase in that area's temperature over time. And as you can see, a lot of it looks quite red here, right? So where we are now, more than 10% of the land surface has been gone up by at least 2 degrees Celsius. And so we're definitely dealing with global warming here. This is a real thing. Of course, science is real and climate change is real. But why this is important is a lot of the theorists believe that once you hit 2 degrees or go more than 2 degrees Celsius of warming, that's when you're really going to start to see the severe climate impacts in our world. And so global warming is part of climate change, right? So if we talk about climate change, what do we think about climate change? Well, this is really the poster child for climate change, right? We like to call this characteristic megafauna of climate change. We have the poor polar bear here, right? Who's really the face of climate change with his sinking, you know, his ice mass loss, right? He's losing his home, he's losing his habitat, right? And this is part of it, right? We burn fossil fuels. This leads to an increased temperature, and this increased temperature leads to ice mass loss. It leads to sea level rise, and definitely shifts in flower and plant blooming, which can have an impact. But today what I'm talking about is the fact that climate change and a warming planet also leads not just to this guy losing his habitat, but there being changes in mosquito habitats, right? As you can imagine, climate change is, and all of these extreme weather events that come with it, which I'm going to discuss today, definitely changes the habitat of mosquitoes and the risk for the diseases they spread, as does warming temperatures, right? And so when you think of climate change, I don't want you only to think of this polar bear. I want you to also think of this, right, by the end of the talk today. And if we look at outbreaks over the last few years, you can see that many of them are caused by vector-borne diseases, right? We've had Zika virus outbreaks, we've had Chikungunya virus outbreaks. And why is this happening? You know, why are these diseases emerging or reemerging? What's actually happening? Well, I think it has a lot to do with the influences of modern life. So we live in a very urban world now, which with it comes, you know, crowding, maybe poor sanitary conditions and so forth, which can lead to some disease emergence with these infections. We change our land. I'm not going to talk a lot about land use change today, but of course we deforest, we reforest, we do land reclamation, irrigation projects. All of these can impact mosquito habitat. Unfortunately, we live in a world that is full of military activities and war. And so any sort of surveillance or control that was going on for these diseases comes to an abrupt end when war breaks out, of course. And so that can have an impact on disease emergence in certain areas of the world. Unfortunately, we have a very limited toolkit and arsenal against these mosquito vectors, and that can have an impact with spread and emergence. And then finally, we live in a very interconnected world, right? You can get anywhere within the world within 24 hours on a plane. And this increased transportation often brings people with these viruses with them to new places. I mean, if the COVID pandemic has taught us nothing more, it's how closely connected we are with everyone on the planet. You know, these borders are really just in our minds. And so we all share infections very easily amongst each other. So what about climate change itself? Well, I'm going to spend a lot of today talking about the warming temperatures and how that impacts the vector itself, and then the diseases that the vector spreads. There's natural disasters and these extreme weather events like floods and droughts and so forth, which also have an impact on vectors, which I'll briefly touch on today. And then this reduced capacity to sustain clean water, which will also come up. So extreme weather events definitely are more frequent. I'm actually from New Orleans originally, and I was talking to a good friend of mine, Julie, yesterday on the phone, and she's saying, oh yeah, here comes another hurricane this week. I mean, anyone who's been alive and paying attention over the last few decades can witness that these extreme weather events, the fires, the floods, the hurricanes, and so forth, are becoming more and more common. And I actually got started as a pediatric infectious disease physician in my fellowship research studying a virus called Rift Valley fever virus. And you may wonder, you know, what is a pediatric infectious disease physician doing being interested in climate change? Well, or climate impacts on vector-borne disease? Well, it turns out that as part of my fellowship research, I was working on this virus, and this virus is incredibly linked and connected to climate, because it turns out that outbreaks of Rift Valley fever are initiated when extreme weather events or large floods from seasonal heavy rains or extreme weather events brought on by El Nino rains and such lead to a lot of flooding of arid places in East Africa. And what happens here is that at the bottom of these little shallow depressions in the landscape, which we call dambos, at the bottom of these sort of pits that collect water are these desiccation resistant floodwater 80s mosquito eggs. And so when the heavy rains come, the eggs are just waiting there. And you go from having no mosquitoes to millions of mosquitoes overnight. I've witnessed this. And the mother mosquito, when she lays her eggs, she actually passes the virus on to all of those eggs. So when those eggs emerge from the floods, they're already infected with virus. And so then those mosquitoes bite livestock, which amplify the virus, and they get sick because Rift is a classic One Health pathogen. And then humans become exposed through mosquito bites or through handling these infected livestock. So you can see that there is, at least with this infection, really it's very intrinsically connected to the climate. And I'm going to talk about other connections over, you know, the next half an hour here with you. And I just want to show you doing work there. This is one of our homesteads actually in this area that's endemic for Rift Valley fever in eastern Kenya. And you can see this is what it looks like in the dry season, and this is what it looks like when it's El Nino rains. And you can just see, you can imagine a lot of ecology is changing here. There may be infections that are really prevalent here that probably aren't going to be so prevalent here, and vice versa, right? So you can just see how everything greens up and the floods come. And with those floods also come Rift Valley fever outbreaks. So a lot of the field work I'm going to talk to you about today comes from a study that we performed, an NIH R01 funded study in Kenya and 4 sites in Kenya. We now have gone on and we continue to do this work, but this particular cohort of febrile children that we followed in 4 sites of Kenya is what I'm going to focus a lot of today's case studies on. As part of this work, we were working in these 4 sites, and we were there to really understand febrile disease in these 4 sites. But along with that, we collected temperature and rainfall and humidity data daily so that we could understand climate at the same time that we were getting information on human disease. And we also trapped thousands and thousands of mosquitoes at all their different life stages at these 4 sites during this 5-year time period. And we were sampling vectors, as I mentioned, in all life stages. So we're getting eggs by trapping for eggs using ovitraps, and we were doing larval and pupal sampling for mosquitoes. And then we're also collecting adult mosquitoes by using backpack aspirators to suck up resting mosquitoes or to lure them to a certain trap called the BG sentinel trap. And we even did human landing catches to understand when they actually came to bite humans. And so we were in a great position because we have both the climate data, and then we have the vector data, and we also have the human data to really try to understand these connections between climate and vector abundance and human disease. And so the first research I want to bring up was actually a Stanford undergraduate student. His name is Cameron Nasrat. He graduated this year. He's now continuing to be at Stanford, hoping to be a physician someday soon. But Cameron was an excellent student and did this project. His project really, the question he asked was, well, how do these climate events really impact transmission in Kenya for dengue? You know, we think maybe the floods will have an impact, but actually, you know, you can imagine the floods come, there's a lot of rain, maybe that means more mosquitoes, but maybe if you have too much rain, it actually washes out mosquito habitats and you have less disease risk, you know, so there are a lot of questions. It's actually quite confusing and nuanced. And so Cameron really wanted to ask, you know, how does climate impact dengue transmission? And so he took our satellite data and also ground truth data on the ground that we're collecting in Kenya, and he classified the months as either flood months or drought months, heat waves or cold waves. And what he did was he took that climate information of those extremes, those climate extremes over 5 years of data, and compared it to average vector abundance and then also human disease risk. And so what did he find? Well, he found that actually floods really resulted in significantly increased vector abundance and generally a higher risk of dengue infection at our 4 sites in Kenya. So it's very interesting, okay? So now we have Rift Valley fever and we have these floods that might be linked to dengue. But I have to tell you that the interactions between climate change and these extreme weather events and these vector-borne disease incidences are actually quite complex, right? And they're very poorly understood because Cameron looked at dengue, but there's been other work done looking at chikungunya. I already mentioned to you both dengue and chikungunya are spread by the same type of vector, a day-biting mosquito, Aedes aegypti, which prefers to breed in man-made plastic containers. Well, these are some of the containers that we found at our sites that actually breed these mosquitoes in Kenya. And during the 2004-2005, there was a very large chikungunya outbreak that happened in Kenya that wasn't linked to floods. It was actually linked to drought. And so how can this happen? You know, it was actually very, very dry time. This was the driest time since 1998 for some of these regions in Kenya. And it turns out that infrequent replenishment of domestic water sources and elevated temperatures at that time might have facilitated transmission. We're going to get to the temperature picture later on in this talk, but for a second, let's think about this, the domestic water stores. So it turns out that because these mosquitoes are container breeders, you can imagine in a drought situation, when you're in an area where people do not have access to safe, reliable, clean water, they're going to collect water and they're just going to store it, and they're storing it in these open containers, right? Well, this mosquito loves to breed in these containers. It's its favorite place to breed. And so what ended up happening is that these containers became really the breeding sites and mosquito factories. And so this is how a drought can actually lead to increased incidence of mosquito-borne disease. But you can see that this is complex. And so when we're talking about this, it's actually nice to have sort of a framework to think about how climate change might actually impact all these different areas of vector-borne disease incidence. And so in order to have disease incidence, I'm going to go backwards here, right? In order to have disease incidence, you need a susceptible host, right? And you need a susceptible host that has come in contact with an infected vector, right? And of course, whether that happens depends on both the host distribution and abundance and the vector distribution and abundance. And climate change, temperature, humidity changes, rainfall changes can impact where a host is found and how abundant the host is, where the vector is found and how abundant and distributed that vector is, and then how often the vector and host come into contact. And also, importantly, temperature in particular and humidity, but temperature, which we're going to focus on today, can really also impact actually the development of the pathogen actually in the vector. And so all of these things have to sort of come together in a perfect storm to have diseases happen. And today we're going to talk about how changes in climate can either increase or decrease that risk. Okay, so back in 2000, there was this paradigm that a warmer world universally meant a sicker world, okay? And this was actually a map that was put out in 2000 showing the risk of malaria transmission by 2020. So by this year, this is what was going to happen with malaria. And as you can see here, I mean, the malaria risk was seen to just multiply in parts of the developed world, including the US, and then all across Europe and some parts of Asia. And you can see that luckily, you know, this hasn't happened. And so, you know, why is this not borne out? Well, this model and many during that time actually were sort of assumed that with every increase degree of temperature, there would just be an increase in how much sickness there would be, an increase in the risk of malaria burden, right? Just literally, the hotter it got, the worse it got, right? But actually, this isn't true because this is really not how mosquito physiology works, right? It actually turns out that for every trait of mosquito physiology that is dependent on temperature, there's actually a temperature optimum at which this trait performs best, right? And if it's cooler than that, it's not performing as well. And if it's hotter than that, we're not at the optimum, right? It's sort of like Goldilocks, too cold, too hot, just right, right? So every mosquito trait has its own temperature optimum, and it can actually get too hot for these things to happen, right? And so this is also how it is with how a warming world is going to impact transmission suitability for vector-borne diseases. You can imagine if we're looking at the relative R0 or the relative transmission risk for any of these infections, it really depends on where you are located, sort of what time of the year it is and how hot it is where you are to see if this 2-degree increase in Celsius is really going to make a big difference for your risk of these infections. You can see if you're down here low on the curve, right, where it's quite cold, a 2-degree increase may not actually increase your risk for vector-borne diseases. But if you're here on the exponential part of the curve around 22 degrees, if you go up to 24, you may actually see quite a big jump in your increased amount of risk for that area. If you're already near the optimum, your risk might not change that much. But if you're past the optimum and you get 2 degrees hotter, it could actually be that your risk for a vector-borne disease actually goes down because it gets too hot for it to transmit well where you are. In order to really start to sort of untangle this, and you can see how nuanced it all is, you also need to know a little bit about mosquito ecology and mosquito preference. And as all of you know, there are many different types of mosquito species that actually lead to vector-borne diseases. I'm talking today a lot about Aedes aegypti and Aedes albopictus. These are the vectors that spread chikungunya and Zika and dengue and also yellow fever. And as I've mentioned, they're container breeders, they're day biters, and they only fly short distances. Compare that with the malaria vector, Anopheles, which also spreads on yang, which is a vector of arboviral infection. They actually breed in very large pools of water, usually ponds, and they fly very far and they feed at night, right? And then there's Culex mosquitoes, a mosquito that we have here in the US quite a bit, spreads West Nile, also Japanese encephalitis and Rift Valley fever. And it likes to feed on animal reservoirs and actually feeds at dusk. And so these different mosquito ecologies actually impact, again, how temperature is going to impact vector-borne disease. So how are we going to figure this out? Well, we're going to use a mechanistic or trait-based framework to really figure out how temperature on its own impacts these host and parasite traits, right, which lead to disease spread. And the way we really measure disease spread is R0. And in general, R0, we call it the basic reproductive number, right? This is the average number of new infections that result from a single infection introduced into a new population. In this way, we're using a sort of a relative R0 because, of course, humans cannot spread vector-borne diseases to each other, right? You actually need the vector, right? These infections are not contagious from human to human. You must have the vector in order to spread them. But we use a relative R0 to sort of figure out how temperature is going to impact these traits and then how that's going to lead to disease spread. And so what are these traits? What are the host and parasite traits that actually determine the value of R0? Well, it turns out that there are so many, as you can see here listed, so many different traits that are going on in that mosquito, right? Mosquito physiology that actually impact the risk for these vector-borne diseases. Now you can see this is a, don't get nervous because I'm not gonna go through difficult math here. Luckily, I work with Erin Mordecai, who I'll show her picture in a little bit. She is my math modeler, biologist, colleague, who I do all of this work with, and she takes care of the math. I like to think about it like this. I'm a picture kind of person. So temperature dependent traits that actually happen, look at this. So just around the mosquito life cycle itself, how many eggs they lay, how many adults die, how well the eggs go into adults, like make it all the way through the life cycle to actually become an adult, and that development of that mosquito, all of those are temperature dependent traits. The biting rates also dependent on temperature. When it's warmer outside, you know, up into their peak temperature, getting to that optimal temperature, they'll actually bite you more. And then there's actually things actually happening intrinsically in the vector itself, right? How competent that vector is for the pathogen, how well the parasite, if we're talking about malaria, or the virus, if we're talking about arboviruses, develops in that mosquito vector is dependent on temperature. And then how infective that mosquito is, how quickly, you know, that virus or pathogen, that parasite gets from the midgut all the way back up into the salivary glands. That's also impacted by temperature. And so our approach, Erin and I's, has been to really, she initially fits all of these physiological responses with data that's collected in the laboratory with the vectors. She calculates the R0 versus the temperature, and then we validate with field data. I'm a field epidemiologist. This is the part that I get to partner with Erin on and actually use our field data to try and validate these ecological models. And then together, we try to project what's gonna happen with future projections of a warming world. Like, how is climate change really gonna impact these diseases? And there is Erin, my partner in crime here, one of my partners in crime, but one of the leads of all of this work. So the first case study I want to bring up is malaria, right? So malaria, as I mentioned, is the most deadly of all of the vector-borne diseases. Still about 500,000 people are dying from malaria every single year. And you can see that the great burden, of course, is in sub-Saharan Africa. So what are the traits of malaria that are actually temperature dependent? Well, you can see here, it's a lot of what I just talked about, right? Biting rates, how often the adults die, how often they survive from egg to adult and so forth. All of these are temperature dependent processes that impact how temperature has an impact on malaria burden. And so what Erin did was she actually figured out that the optimal temperature for malaria transmission was a lot lower than previously predicted. So initially people thought, oh, the perfect temperature for malaria transmission is around 31 degrees Celsius. Well, Erin actually looked at all of those unimodal curves for all of the different traits that I just showed on the last slide, and actually used empirical data and a lot of lab data from the vectors themselves, and actually was able to validate her new estimate of malaria transmission, which is seen here in blue, with all of that field data, really validate it and show that actually the perfect temperature for malaria transmission is 25 degrees. And what this means, you may be saying, well, 25 degrees, that's about 77, does this really matter? Well, it turns out when you're trying to predict how climate change is going to impact this, it's actually really important to have the correct temperature optimum for these different infections. And so this was, again, all based on vector data, but it had never been really taken to the clinical side, to humans, to really ask the question, is the temperature effect predicted by Erin's ecological model really applicable to clinical malaria incidence, right? Is temperature having an impact on whether or not we're seeing kids sick with malaria? And so this brings me back again to our 4 sites in Kenya. So as I mentioned to you, that we were enrolling children with fevers at these 4 sites over those years. And so along with the climate data and the vector data, we were getting information with children with undifferentiated febrile illness. And so we have about 6,000 children that we enrolled over those 5 years in these 4 sites in Kenya, 2 on the coast of Kenya and 2 on the shores of Lake Victoria on the west side. And so our malaria smear positivity at these 4 sites is actually quite different, right? So of all the febrile children coming in all over those 5 years, on the coast in our urban site in Ukunda, about 50% of kids were positive for malaria, but only in about 60% on the rural site. But when we looked on the west side of Kenya, at our rural site in Chilaimbo, more than 80% of the kids were smear positive with fever there, and about 60% were positive in urban Kisumu. And you can see here just the temperatures of these places over those 5 years. You can see that Chilaimbo and Kisumu are a little bit colder than on the coast. On the coast, it's a little warmer. And so I had Melissa Shah, who is a postdoc in my laboratory, who was funded by an MCHRI postdoctoral support fund, and she was able to do this work. So what Melissa did was she actually tried to use our clinical data in those 4 sites to really see if we saw an effect of temperature on smear positivity. And so she calculated the mean temperature, the effective mean temperature, which is here on our x-axis, and smear positivity across our 4 sites over those over all of those years. And she controlled for a lot of things that might be important for why you're coming in with febrile disease, right? She controlled for rainfall, she controlled for bed net use, for sex, for age, and for socioeconomic status. And shockingly, her data in the clinics completely mirrored what Erin was seeing in her ecological model. So you can see here the smear positivity rate peaked at a temperature of 25 degrees Celsius. So this was really, really cool to see empirical data and ecologic data actually completely align with what we're seeing in the clinics. And Melissa was able to publish this MCHRI funded work in a paper last year in Parasites and Vectors. And so when we broke it down by the 4 sites, remember that Chilaimbo, that rural west side site, was the site that had the highest malaria smear positivity. And you can see here that as the temperature got closer to 25, you know, the incidence in Chilaimbo peaked. And then as it got a little bit warmer in Chilaimbo, again, it went down. And so Chilaimbo was probably one of our hottest areas for malaria smear positivity, because it really was around 25 degrees a lot of the time. And as you can see in our other sites, especially on the coast here with these orange and red dots, you can see it almost got too warm for malaria smear positivity. And you can see that this clinical data actually mirrors the ecological models very, very well. So what does this mean? We're just working in 4 sites in Kenya, right? What does this mean for the rest of Africa? What does this mean for the great burden of malaria? Well, it's not so much that malaria burden is just going to increase everywhere in the world as the temperature warms. It actually means that, you know, these nuanced shifts in temperature are actually going to shift the burden of malaria. It's going to shift in geological space, and then also perhaps in time about when people are going to be most at risk. You can see right now, you know, we have a lot of Central and West Africa, which are being hit hard by malaria, but as time goes on, you can see that there will be a shift in the burden of malaria eastward, and so that some places may end up being too hot for as much transmission as we're seeing now. So a shift, it's a shift. Well, what about the other infections? What about dengue and chikungunya and Zika? The mosquito virus infections? Well, again, as you'd imagine, Aedes aegypti, which this is the worldwide distribution of Aedes aegypti, and the sister species, Aedes albopictus, which both can spread dengue and chick and Zika. You can see each one of these mosquitoes, each species has its very own little curves when it comes to how they develop and all of these temperature dependent traits within the mosquito, right? And so Erin went and looked at all of these curves for Aedes aegypti and Aedes albopictus, and she was able to make a curve for these mosquitoes. So we showed it to you for malaria, right? Malaria peaked around 25 degrees, while Aedes aegypti, this darker blue here, is the dengue mosquito. And you can see the dengue mosquito likes it hotter, right? It peaks around 29 degrees. And Aedes albopictus, which is a sister species, which has been responsible for spreading chikungunya all over Europe, likes it a little bit warmer than malaria, but cooler than the Aedes aegypti mosquito. So it peaks around 26 degrees. So it turns out every single mosquito species has its own curve. And it's important to know that when you're trying to figure out how climate is going to impact these diseases. You can see here Erin's model for Aedes aegypti in the dark blue color again, and it lined up with a lot of other previous models, including other validated models of Aedes aegypti, or the dengue vector in the world. And so we go back to our 4 outpatient sites in Kenya and we ask the question, well, what about all the kids without malaria? You know, when did their temperature, you know, how hot was it outside when that was peaking? And so as I've mentioned, you know, we have this dengue curve now, which peaks at 25 degrees Celsius, right, which is about 77 degrees Fahrenheit. And then we have the dengue curve, right, for the Aedes aegypti mosquito, which peaks at 29 degrees, which is 84 degrees Fahrenheit. So when we went and we actually plotted our non-malarial fever temperature, you can see that, again, it mirrors quite well with this dengue curve, right? Peaking, continuing to peak as the temperature rose, right? And so when you actually put on the malaria febrile illness and then the non-malarial febrile illness, you can see there's definitely overlap, but these clinical data really do validate the ecological models that temperature really might be having an effect here. And so what does this mean worldwide? Well, again, as the temperature warms, right, we're looking here at months suitable for transmission of dengue. It's just going to be that much of a greater portion of the world is actually going to be suitable for dengue transmission on a year-round basis, right? It just extends where there is potential risk for climate is allowing these mosquitoes to be able to transmit these diseases. Of course, in order to actually transmit them, you need the vector itself and you need the virus or the pathogen within that vector. But still, we're talking about possible risk here for transmission of these infections. Okay, so when you look, this is work that Erin has done recently. Again, every single mosquito species, right, has its own curve. And so if you're on this, and depending on where you are in the world and what your temperature is, right, maybe if it warms 2 degrees, these infections are going to go up. Or maybe if you're already where it's hot, maybe these infections are going to go down. Or maybe if you're in the middle, it's going to stay about the same. But you can see that it's quite nuanced, right? It's not just warmer world equals sicker world. And so what about malaria and dengue in Africa? So I've been working on both of these, like I mentioned in our fieldwork in Kenya. And so we wondered, wow, well, what is climate actually gonna shift and promote a shift from malaria disease incidence in sub-Saharan Africa to dengue? And so we recently just published this paper in Lancet Planetary Health where we talked about malaria suitability and how that's going to change over time with current climate predictions, and dengue suitability. And as you can see, as the world warms, more and more of sub-Saharan Africa is going to be at risk for these Aedes aegypti vector diseases. So again, back to the mosquitoes. So the Aedes mosquitoes, like we've been talking about, they love 29, right? The malaria mosquito is a little bit cooler at 25, and the Culex mosquito likes it even cooler, around 23 degrees, right? And so again, asking the question, how is climate change really going to shift disease burden? It depends on the mosquito, because the disease, of course, is vectored by each one of these different species, and different vectors actually vector the infections in different parts of the world, right? So you need to know what the temperature is where you are, which of these vectors actually carries the diseases that you're looking for, right? And so it is very sort of nuanced, and it's quite complex.
Desiree, 2 minutes.
Okay. What are the challenges here? Well, we have a non-immune population. Really, we have widespread competent mosquito vectors. We don't have any rapid testing, and we really have limited physician knowledge. Poor diagnostics and no treatments or vaccines for these infections, right? And so I'm going to, we've done work in these vectors trying to understand really how we can mechanistically link mosquito abundance and whether or not weather suitability can actually drive mosquito abundance, which drives mosquito diseases, right? Mosquito vector diseases. And so Jamie, who is a postdoctoral fellow in our lab, did this work. And I will just show you that she took climate inputs and then modeled how many mosquitoes we should see and how many disease cases. And we did this work in Kenya and also in Ecuador, which both have transmission of dengue. And Jamie was actually able to use models to predict the mosquito vectors at all the different life stages, and even in 2 different countries, right? She was actually able to predict in Ecuador how many mosquitoes we would have, and in Kenya, and she was able to predict disease. So we're actually able to use climate models to actually predict when we're going to see all of these different mosquito vector diseases in our communities. And this work is actually under review at Nature right now. And so you can see here that predicting this stuff is important for knowledge, but really what the people on the ground need is actually actionable information to be able to use limited vector resources to go out and kill these vectors, right? And so Jamie was actually able to make an app to share with our public health partners in Kenya to actually know when they should tackle vector control for these infections. But I've talked about climate today. I just want to, end the end here, just say human behavior also matters. Of course, climate is not the only thing that's important for determining when and where conditions are suitable, but clearly it has a big impact and it exerts a nuanced effect on these vector-borne diseases, depending on the location of where you are in the world and what your temperature is there. I've shown you that climate actually can shift the burden of these diseases from malaria to arboviruses in many parts of sub-Saharan Africa, and the degree to which climate actually translates into changes in disease risk depends on not just on climate itself, but there's a lot of other factors, right, that I haven't discussed. You know, herd immunity and whether the people have seen this pathogen in the past and whether they're protected. Of course, your housing type, how often you come into contact with that vector control, public health efforts, which are trying to decrease these diseases, and then of course humans, right? Now, we talked a little bit about vector ecology and vector behavior, but of course humans are mobile. They have behaviors that either put them at risk or protect them for these diseases. And so that of course has a lot of impact as well. So this is just the picture of my team in Kenya, which made this work possible. I'm here as the spokesperson, but there are a lot of people who make this sort of work possible. I've mentioned Aaron Mordecai, Jamie, and then also Eric Lambin, who worked on the Woods Institute funded project that supported Jamie and being able to predict both vector abundance and climate impacts on disease in Ecuador and Kenya. And I want to thank, again, MCHRI, who supported Melissa to do a lot of this work, my fantastic team at Stanford, and then also the Woods EVP and our NIH grant, which I share with Bryson and Dengue and Francis Matuku, who are my two Kenyan leads, and a lot of our collaborators who make this work possible. So thank you very much for your attention, and I'm happy to take any questions.
Well, thank you very much, Desiree. This is really eye-opening and very practical in a way that you can predict the upcoming infections. We have a lot of questions from the audience. And so I'm just gonna try to group them into different categories and so one of them is a basic one, which is what is the risk of, actually is, how do you describe how the bite by the vector transmits the virus to the person? Just a kind of a basic level question first.
Mm-hmm. Absolutely. So what usually happens is, so the mosquito itself gets infected. Either it's infected by the mother mosquito and was already born with the virus, or it takes a blood meal from an infected person. And what happens is that virus is in the blood of that infected person, and then the virus goes into the mosquito, right? And that starts off the infection within the mosquito. But in order for the mosquito to actually be infected, the mosquito has to swallow that blood. It has to go through, to the ovaries and then back out into, from the midgut back into the saliva, so that then the mosquito can go and bite an uninfected person and transmit that virus. Now, with these infections, it depends how many bites you need in order to become infected, right? Usually with malaria, I think it depends on how much pathogen, like how much malaria is in the saliva of that mosquito, or how much virus is in the saliva of that mosquito. But usually people say maybe around 4 bites or so for malaria in most places will get you malaria. For the viruses, it also depends, right? It depends on how infective that mosquito is. But what happens is then the virus goes into you, it goes through your immune system, it goes into your lymph, it goes into your white cells. These viruses love to infect, they infect the endothelium, but they infect a lot of immune cells, and then you get a viremia, and then a few days later you end up with your symptoms.
Right. think that's really helpful. So a lot of questions on viral adaptations to the temperature. And so questions such as, what's the risk of mosquitoes and the viruses that carry adapting to the warmer temperatures, mutating in such a way that they can, is there evidence for that kind of adaptation?
Absolutely. So these, so the mosquitoes themselves can adapt. So the vectors can actually adapt, right? You can imagine, I'll give you one just little study of Kenya. So in Kenya, malaria is spread by Anopheles gambigambi. But there's a subspecies called Anopheles gambifunestus. And so the difference between these two vector species is when they feed. So there have been huge efforts for public health control, right, of malaria in sub-Saharan Africa. And the main one has been the distribution of insecticide-treated bed nets. And so this works because these vectors bite at night. And so you get under your net and you're protected from the vectors from Anopheles gambi coming to find you. But it turns out that Anopheles gambigambi is no longer really a huge malaria vector in sub-Saharan Africa because it's been taken over by Anopheles gambifunestus, because it turns out funestus bites a little sooner, bites a little earlier in the night. And so it gets to people before they're under their net, right? So yes, nature is always, it's incredible, right? Nature is always changing. Can the vector adapt? The vector can adapt its behavior and its biological processes over time, right? To be able to deal with these changes in temperature and changes in habitat, and they are doing that. And we have many instances where the virus, as the question's asking, the virus can also adapt. So Chikungunya virus adapted. So there was a mutation in the virus at region 226 that allowed it to actually be more infective for Aedes albopictus mosquitoes. And so normally chikungunya is spread by Aedes aegypti, but now it was spreading really well in Aedes albopictus. And so what ended up happening is we had outbreaks of chikungunya in Europe, especially Italy, because there's tons of Aedes albopictus all over Europe, all over southern Europe. And now you had a virus that was well adapted to that new mosquito species. So absolutely, adaptation is going to be happening along with this, right? We can only predict what we can predict now and then take in more knowledge as we get it to understand, and make our predictions even better in the future.
Right. And you'll have to actually probably get not only just viral adaptation, but also mosquito adaptation. And then a sort of next level of question which came up, which is the level of mobile individuals, because many of the people in Africa are relatively sedentary, but I mean, they don't move as much. But now what about as they move into a higher class where they're actually now more mobile? How do you figure that into it?
Oh yeah, absolutely. I mean, so at least my partners and friends in Kenya are highly mobile, right? They move a lot in country and out of country. And so, yes, absolutely, because these infections get moved with people who are sick with them, right? So, we carry the virus in us. We may or may not have symptoms even when we have the virus in us. Like, for instance, dengue, only about a quarter of infections are actually symptomatic, so you feel sick with it. Most of the time, people have the virus and are able to infect mosquitoes, but don't even show any signs of symptoms. They have no sickness. And so they move around like they would normally, and they're bringing the virus with them. So if the vector happens to be there and feeds on them, they can absolutely be that index case and start a little outbreak of whatever that virus is there. So human mobility is huge. We're trying to look at human mobility in our work in Kenya to try and understand how both social networks and what we call activity spaces, which is where you spend your daytime hours, right? These mosquitoes bite in the daytime. So you're not getting infected at home, you're getting infected at work or school or, your church or mosque. And so human mobility is also definitely linked to risk of these infections.
Yeah. And then for human, basically bites, one question was about, they have two children and one child gets bitten, and doesn't. Is there like a universal lotion for kids? But also, why does one kid get bitten and doesn't?
Yeah, so it turns out that mosquitoes can sense over 100 proteins on their little antennas. And I don't know about you, but like mosquitoes love me. There are some people that mosquitoes just love.
I'm very lovable too.
And then other people, they never bite, right? So it turns out that some of us are just really delicious to these vectors. And, they use their antenna to sense us out and to find us. and to bite us. And so it could be that in a certain household, you know, you have several children and some of them are just sort of the mosquito magnet, right? Scientists are trying to figure out why that is and how to prevent that from happening, right? I mean, you can imagine if we could isolate whatever it is that they love in us and use that as a lure, we could get rid of them and they'd stop biting us, right? And so right now, as far as repellents and things like that, to protect yourself, you know, there's the ways that you can protect yourself from these mosquitoes. In particular, the first important way is actually something we call larval source reduction, which is cleaning up your area, making sure that mosquitoes aren't breeding in your dog dishes and your flower pots in your backyard, right? That's step one, right? A clean environment where there's no containers for these mosquitoes to breed. Because once they find you and they find a little place to breed, they'll never leave you. They'll just keep feeding on you for their entire lives. So cleaning up the environment is big. And then also, of course, you want to protect yourself by wearing personal protective, you know, dressing. So of course, long sleeves, long pants, things like that. And then mosquito repellents, right? I always use DEET, which is effective. The more percentage, usually I use around 30% DEET. It protects for about 5 hours or so. For now, all we are able to do is really clean up the environment and then, you know, use these sort of repellents to get the mosquitoes to stop feeding on us.
Right. Great. So then the flaviviruses are single-strand RNA viruses, sort of like COVID, SARS-CoV. So with all this hype about vaccine technology for SARS-CoV-2, what's the status for vaccines, or why isn't there a better vaccine for these more like these virulent viruses that kill a lot of people?
Right. So there's a lot of work, right? People have been working on malaria vaccines for a very long time, right? It's a very complex parasite. And so it's got a very complex life cycle in the human. And so many people, and there are many vaccine trials, many people are trying to work on a safe and efficacious malaria vaccine. As far as dengue and chikungunya, again, for chikungunya, ever since chikungunya, there's large outbreaks and a lot of people across the world got sick when it came to the Western Hemisphere and then also into Europe. There are many people who are also working on chick vaccines. I'm hopeful that we will have an effective chick vaccine sometime in the next 5 to 10 years. And then as far as dengue goes, there was a dengue vaccine that was released, but there was some difficulty. Dengue is particularly tricky because there are those 4 serotypes of dengue and you need to protect well against all 4. Because as we know, people who have one exposure to one of those viruses, when they get their second infection, they're more likely to get severe dengue. So you definitely don't want to give a vaccine and then actually only protect against 1 or 2 of the serotypes of virus and actually put people at more risk of severe disease. You must protect against all 4. And it can be very tricky to do that. And so there's so much ongoing work on dengue vaccines. And I also expect soon that there would be an effective dengue vaccine.
Great. So there are a lot of people that want to know, how can they read more about the research, more about also predicting climate change and their risk for, could you give us a few, I know we can send out on our MCHRI website, we can basically post some things, but but just for generally, where would some people can read more about this very interesting topic?
Oh, wow.
Besides your lab website.
Right. Yeah.
Which would be a very good choice.
Right. I think, there are a lot of journals now, this is like a movement, right? So we're part of a movement. We're part of a planetary health movement. And Stanford actually is right in there at the front of this movement because there's a lot of motion and energy and expertise right now really sort of coming around this notion of planetary health and creating a center for planetary health at Stanford. Because, climate change and vector-borne diseases is one tiny little aspect of how climate change is going to change the world, right? I mean, there's food and crops and all sorts of diseases that aren't vectored by mosquitoes and so forth that have an impact on us. I mean, we are completely as human wedded to the health of our planet, right? That's why the name of my talk was Healthy People, healthy planet, healthy people. You can't separate us from our planet. And so this whole movement, there's more and more happening. So I would say there are journals now, like the Lancet Planetary Health, where Aaron and I just got this paper published. That journal, if they're interested in scientific reading about planetary health, that is a great place to start. If you just, there's going to be a Center for Planetary Health at Stanford coming up here, the Woods Institute at Stanford, and their website is a great place to start and start reading about these infections. Same thing with the Center for Innovation in Global Health at Stanford has a huge just a huge interest and push towards planetary health. And so I think there's going to be more and more, and anybody who's interested in this, please get involved with it. I think it's crucial for both the health of us, the health of all the other animals and plants on the planet, and the health of our planet itself, for us all get together and solve these issues and be able to both predict what's going to happen with climate change, but also prevent the harmful impacts on all of us.
Well, you now have us very excited about getting involved about the Healthy Planet Initiative. And so thanks very much. We really appreciate this inspiring lunchtime seminar series talk. And for those in the audience, please reach out to Desiree. Look, her website, also the MCHRI website has information, and also stay tuned for additional, but most involved, get involved with it yourselves. So in all ways. So thank you very much again for this wonderful presentation, and look forward to the MCHRI Symposium next month. Thanks very much, everybody.
Thank you. Bye-bye.
Thanks.